Katrina had a poster at ASCO GU, which is a large medical conference for oncology where researchers and doctors share kind of the latest developments in cancer research and clinical research, bringing more treatments to patients. And so the poster session that was at that conference is kind of like a medical science fair where researchers and doctors come and present their work. It may not be published work yet. A lot of times it's while they're still gathering the data for their paper, a way to exchange ideas. So not everything that's presented in the posters always pans out. It's really a way to exchange early ideas. And then there are some posters that are published. So we'll touch a little bit on kind of where in the process a lot of this work that was presented in the poster is, as Katrina shares with us today. But just with that context in mind for what these poster sessions are at ASCO GU, we welcome Katrina, who will be sharing with us a little bit about her poster and the research that she brought to that conference.
Thank you for having me, Katie. Again, my name is Katrina. I'm a fourth-year medical student, and I work in Dr. Braun's lab here at Yale. So a little bit of background about why we did this work and did this research. So Chromophobe RCC is the third most common subtype of kidney cancer. It accounts for about 5% of all kidney cancers. The most common subtype of kidney cancer, and the one that has been studied by far the most, is clear cell renal cell carcinoma. Unfortunately, the treatments that we have that are what we consider the gold standards for clear cell RCC don't seem to be very effective at treating chromophobe RCC. So those are things like immune checkpoint inhibitors. And actually, more generally, we just don't have a very good idea of which of the existing treatments are the most effective for chromophobe RCC. And also, of course, we're really interested in developing new targets that we can use to treat chromophobe in the future. So that was kind of the motivation behind our poster. With that in mind, one of the things we were interested in looking at was really understanding the biology of chromophobe tumor cells. And so to understand the tumor cells, we take it a step back and actually look at their cell of origin, or the cell that these tumor cells derive from. So that had been established for clear cell renal cell carcinoma and had been studied to some degree in chromophobe, but hadn't really been established with a technology we call single cell RNA sequencing. So that was what we used in our study. On the poster, what you can see is that here in figure 1A, we have a schematic of the nephron in healthy adult kidney. And so you can see that there's lots of different parts that make up your kidney. And what we were looking for was which of these parts the chromophobe tumor cells actually come from. So what we did is we looked at tumor cells, that's down in B, the tumor cells are in purple, and then healthy cells from each of these different parts of the kidney. And then we were able to compare those one at a time and see which of these healthy cells was the most similar to the chromophobe tumor cells. And so that's what you're seeing here in C is actually their similarity index. And so what we came up with was both in our own cohort of chromophobe RCC tumors, and we used an external validation cohort kind of to confirm these findings. The alpha intercalated cells, which are denoted ICA in the healthy kidney, are the most similar to the chromophobe tumor cells. And if we were looking at this for clear cell RCC, the cells labeled PT, which stands for proximal tubule, would be the ones that would be most similar to clear cell. So completely different parts of the kidney that these two tumors are actually deriving from. Then looked at some clinical data from the International Metastatic RCC Database Consortium, or IMDC, and evaluated a current drug class that we use to treat renal cell carcinomas called mTOR inhibitors. And we looked at that in metastatic chromophobe, in metastatic clear cell, and compared how it did in each.
What was the most significant finding when you were comparing those as far as the response? And also, what made you look for mTOR specifically? Was there something that was different between those two cells that really highlighted as mTOR being the main pathway to look at?
Yeah, so once we had done that cell of origin analysis and come up with that alpha intercalated cell, we directly compared the genes expressed in the alpha intercalated cell and the chromophobe tumor cells. And there were a couple of different genes and gene pathways that were really different between those. So One of those was ferroptosis, which is a form of regulated cell death. And we actually found several genes that were different between the tumor cells and the healthy cells. And then we also found changes in a pathway called mTOR C1. And that's the pathway that's targeted by these mTOR inhibitors, which is what led us to look at that clinical data specifically. I can talk a little bit more granularly about that clinical data as well. The patients who had metastatic Chromophobe RCC who received first-line mTOR inhibitors, they had better overall survival than those with metastatic clear cell RCC that received the same first-line treatment. And this really aligned with our finding that the mTOR C1 pathway was altered in chromophobe tumor cells.
So you looked at this database, and this was a database of patients that had been previously treated. So kind of looking back retrospectively, is that correct?
Yeah, so this is a retrospective study. So these patients, this was the first treatment they got, but it was treatment that they had received in the past. So before we're actually sitting and looking at it, we're looking back at the treatment they've already received. I'm glad you brought that up because with retrospective data analysis, there are always going to be some limitations. And so we would love to do some like prospective trials, which means we assign the treatments and then can look at them going forward. And that helps really confirm some of these findings.
How many patients were looked at in between clear cell and chromophobe in that data? Do you know?
So because clear cell is more common, we had more patients for the clear cell cohort. So we had 292 patients for clear cell and we had 32 patients for chromophobe.
Where do you see the next steps for the research and also kind of where is this research in the process? We talked a little bit about that poster sessions can be sometimes some preliminary research. Where is this research kind of in the current pipeline to being more widely available?
Yeah, so you have great timing. We actually just got informed that the paper, that this research is a part of, been accepted for publication in the Journal of Clinical Oncology. So hopefully this will be available, both the information that was presented in this poster and then a lot more detail about the immune landscape of Chromophobe RCC and more clinical data as well, looking at both immune checkpoint inhibitors and other targeted therapies. So hopefully that will be published in the next couple of months and will be available for people to read and to learn from. And then for your question about future directions, as I've kind of alluded to already, I think really what we need right now is to deeply understand the biology of chromophobe as a unique disease. And based on that biological understanding, we can then rationally design treatments that really target chromophobe specifically. So a couple of those things, for example, we have both tumor intrinsic characteristics. So one of those, for example, that we found in this poster was the intrinsic resistance that these tumor cells have to ferroptosis. So one big next step for that is going to be looking at existing drugs and maybe even some novel drugs that can induce ferroptosis in these tumor cells. There's some preclinical studies that are looking at some of these drugs in cell lines derived from chromophobe RCC tumors, and these have shown promising results as done by Dr. Henske's group. So obviously from that preclinical cell line study to coming into the clinic, there's going to be a lot of work to do to make sure that treatments are safe and looking at side effects and things like that. But I think that's one very interesting avenue that we're currently looking at.
Can you explain in simple terms what ferroptosis is and why it might be important for chromophobe kidney cancer?
So ferroptosis is actually a relatively recent discovery, actually from 2012. And it's a form of iron-regulated cell death. So this is different than other forms of cell death like apoptosis that we more commonly talk about in the cancer world. So ferroptosis involves the accumulation of iron in your cells. And the iron accumulation creates these little volatile molecules known as reactive oxygen species. These molecules react with things like proteins, DNA, fats in the cell and cause damage. So more specifically for ferroptosis, there's a process called lipid peroxidation where these reactive oxygen species react with the lipids in your cell membranes, both around the cell and throughout the cell. And eventually, if there's enough damage from these reactive oxygen species, the membranes start to break down and it causes the cell to die. So for chromophobe specifically, we're seeing that these tumors are expressing genes that protect them from ferroptosis. So if they are not being regulated in the way a normal cell would, where if something is going wrong, the cell should die, it contributes to the growth of these tumors and their survival in our bodies. And this is interesting for us to understand because if we can induce ferroptosis in these cells or disable the mechanisms of resistance that these cells have to ferroptosis, we can use that to target and kill these cancer cells. And as you know, we are in need of new targets for treatment in Chromophobe RCC. And then speaking more to some of the stuff that's coming out in the paper, the immune landscape of Chromophobe looks very different than the immune landscape of clear cell. There's very low immune cell infiltration in Chromophobe tumors. The T cells that are in the tumor are not really specific for the tumor cells. They don't seem to be very active. So addressing that is also going to be an important factor for considering treatments in the future.
That's awesome. So it kind of sounds like this paper is really kind of laying the foundation for we've just been using treatments kind of very broadly for kidney cancer, but now this is really laying the foundation for us to have more treatments that are more tailored to chromophobe kidney cancer. I know this has kind of been done in other subtypes, like clear cell has VHL and the new target with belzutifan. Is that a future direction that kind of this work is aimed at, is being able to help kind of find similar things more tailored towards chromophobe?
Yeah, it's a great example. It's a very similar idea. So by really understanding the biology of chromophobe, understanding what things are dysregulated in that process from healthy cell to tumor cell, and then targeting those changes, we're really able to create a treatment that is specific for the tumor and hopefully has also less side effects on healthy cells. So that's really the goal with this study, both this poster and the upcoming paper.
It's exciting work. So thank you for sharing with us today. Do you have any final words you'd like to share with the chromophobe kidney cancer community or any patient that may be listening in?
Yeah, I think it would just be important to note that a chromophobe RCC, while historically has not been studied as much, there's a lot more research being done in the field about chromophobe specifically. And as we've discussed, this is going to be really helpful for creating new and very tailored treatments. I also mentioned that the studies we've done shows that some of the existing treatments, such as the mTOR inhibitors, do target chromophobe RCC, that there are definitely biological pathways that we have identified that could be targeted in the future. Of course, this research takes time, but we're working on it, and hopefully the growing understanding of the biology of chromophobe RCC is going to translate into better treatments and better outcomes for patients in the near future.